This lecture on overview of recombinant DNA technology is part of the lecture series in basic molecular biology. What is recombinant DNA? For this, I would take a classical example of insulin, which is a protein, and gene for insulin is present in all types of cells, but those are expressed only in the cells of beta cells of pancreas. So we isolate DNA and you take out the gene by some method. Now this gene can be transferred to some vector, such as a plasmid vector. For example, this is the human DNA and this is the gene for insulin that is isolated, that I will discuss later how you can isolate a gene. And then you put that gene in some vector. There are different types of vectors that I will discuss later as well. So plasmid is one of the very basic types of vectors. So we put our gene by DNA ligase in this vector. Vectors are circular in nature, so we have to open this circle at some place by restriction enzymes. And when you open this circle, then you can put this gene of your interest. For example, in our case, insulin gene, you can put over here. Why we need this vector? Because we want two things. One is the cloning of the gene and other is the expression of the gene. Cloning means you get identical copies of their gene, and expression means you get transcription and translation of the gene. Simple cloning can be done by in vitro PCR, but for the expression of the gene, one needs some system where this transcription and translation takes place. And this translation especially is a complicated process because it requires 20 amino acids and ribosomes, messenger RNA, transfer RNA, and there is a very complicated machinery which bring about this translation process. You can see one of my other talks on transcription and translation. So if we take this gene directly to some living system, for example, this is a bacterium. Here we take the example of E. coli. Consider this is an E. coli cell. This is called as host. So we take this gene into this host. The process is called as transformation. But if we transfer this gene directly into the host, what would happen? These cells, bacterial cells, have their own defense system. So they will take it as a foreign DNA and chop it up. So we transfer this gene into some vector and then we transform this vector into this host cell. And this vector has certain properties that it cannot be chopped up by the host cells. Now, once inside the cell, when you grow these cells, you get multiple copies of this plasmid DNA. And in this way, you get cloning of your gene when this plasmid, which is being replicated independently, independent of the bacterial chromosome. This is the bacterial chromosome, and you know in bacteria single chromosome is there. So plasmid copy number will be high. It may be few to few hundreds or even up to 3000 per cell, depending on the nature of the plasmid. So you can grow these. When you grow this, you get a large number of these plasmids, and your gene of interest is also cloned, meaning you get multiple identical copies of the gene when this plasmid is replicated. Now coming to the expression of the recombinant proteins. As in the last slide I mentioned how you can do the cloning of the gene. If you put that gene in a vector which contains signals for transcription and translation, you can express that particular gene into protein. and those transcriptional signal are the promoters, operators, and the transcription start and stop sites. And the translational signals, they include ribosome binding site, start codon, stop codon, and all that. So when we transform this expression vector, this is called as expression vector, into a host cell, and we grow this, along with this cloning, you also get these two processes, that is transcription and translation. Ultimately, you get these proteins, and these proteins, which are made by recombinant DNA, these proteins are called as recombinant proteins. Now, first we will discuss how you isolate a gene. Gene isolation had been a very tough job, and people spent years of their lives on isolating a single gene, because the size of the DNA is very large. Like in human DNA, 3 billion base pair in the haploid human genome. And if you get a job that you have to just read the human DNA sequence, you come in 9 in the morning and you leave by 5. So 9 to 5 you have the job to count the bases ATGC, ATGC. It will take 80 years to complete it once. But the size of the gene is very small as compared to the size of the DNA. It is just a few hundred or few thousand base pairs. So isolating a gene from this huge DNA is just like you have a big stack of wheat straw. For example, you have a big hall and it's full of wheat straw, and you drop a pinhead in that wheat straw, and you have to find that out. This used to be the case for the isolation of a gene. But thanks to the molecular biology, especially the polymerase chain reaction, it brought in revolution. Carey Mullis, who invented this polymerase chain reaction, he did that in water baths, of course, because there was no machine, but he invented that, and this is a thermocycler, one type of machine for the PCR reaction. So PCR, now with the help of PCR, you can isolate gene very fast, maybe in a 1 day or even less. The PCR is an in vitro method for enzymatic synthesis of specific DNA sequences, and it uses two primers that hybridize two opposite strands. How this polymerase chain reaction has 3 steps, because you know the DNA is double-stranded. So first step is to separate these 2 strands, and this is melting of the DNA or denaturation of the DNA that can be brought about at 95 Celsius. After this denaturation of the DNA, the primers have to bind to specifically to the DNA sequence. Specifically means if you want to isolate gene for insulin, you need primers that are complementary to the gene for insulin. So these 2 primers will bind to the separated strands of the DNA. So this is called as this step is called as annealing of the primers, and the temperature for this step depends on the melting temperature of the primers and this DNA, that how good they bind to specifically to the DNA template. And the third step is the amplification, basically is the DNA polymerization, and this takes place at the 72 degrees Celsius with the help of DNA polymerase. Normal DNA polymerases, of course, cannot function at this high temperature, but there is a specialized DNA polymerase which is called as Taq polymerase. Taq is from Thermus aquaticus, a hot spring bacterium from where this tag DNA polymerase was isolated. So nowadays, most commonly this DNA polymerase is used, although there are thermostable DNA polymerases from some other bacteria. Those are also being used, but this is the most common one which is used. So this is called as one cycle of PCR: denaturation, annealing, and extension. So this is one cycle. So here it is more clear, the three steps. Here is the denaturation. Double-stranded DNA is denatured, then annealing of the primers at specific place, and then extension that requires DNTPs and DNA polymerase. So basically it is just a replication. And you know from replication that DNA polymerases cannot initiate new chain. They can extend already made chains only. That's why a primer is needed in DNA replication in the cells naturally. You can have my lecture on DNA replication for further details, but these are the primers which are required that have to bind to this DNA. And then the extension. This 3' end of the primers has to be very specific because this determines the specificity. If this end is not specific, you won't get amplification of your desired gene, because this is the place where tag DNA polymerase binds and extends this chain from in this direction. So this has to be very specific. This DNA amplification by PCR is an exponential process. You can see this one DNA, then there are 2 copies of DNA, then there are 4 copies, 8, 16, and so on. And only in 35 cycles, you get 34 billion copies of the DNAs. And these 35 cycles can be completed in a PCR machine in just less than 3 hours. So you can get so much quantity of DNA, 34 billion copies in just less than 3 hours. Now there are certain modern machines which are further reducing this time, and they are trying to make it to minutes rather than hours. Key to the successful amplification of your gene by PCR is the correct primers. You can design your primers by using various bioinformatics softwares. Here I will go through just a very fundamental principle of these primers. Usual amplicon length is between 250 to 1000 base pair, but as you know, you can have the longer amplifications as well. The primer should be unique. 18 to 28 bases should be there, and melting temperature, the temperature at which half of the DNA is melted, ideally it should be more than 55 Celsius, and 50% GC composition of the primer is recommended, and there should be GC caps at the 3' end of the primer. As I mentioned in the last slide, that the 3' end of the primer is more important and should be specific, and this GC cap at the 3' end, that will help in tag DNA polymerase to initiate the chain in quite stable form. If there is an AT at this region in the 3' end, what may happen is that there may be breathing of the DNA, meaning AT having 2 hydrogen bonds is not as strong as GC having 3 hydrogen bonds. So 1 G here, 1 C here. So this GC will allow the stability of 3' end of the primer to the DNA template that will help the tag polymerase to initiate this chain. There should not be any internal complementarity within the primer itself. Otherwise, what would happen? Primer will self-complement with each other rather than binding to the DNA template. And there should not be any primer dimers, meaning two primers should not have the complementary sequences as well. Otherwise, these two primers, they are shorter in sequence, they will complement with each other and they won't bind to the template. And in most cases, it's better to have purified primer, like it can be purified by HPLC, but this is not essential. But if your PCR is not working, you can try this. So the core thing in PCR is the correct primer. In many cases, especially in eukaryotic cells, there are long introns. Rather, intron size is much longer as compared to the exons. And if you want to express that gene, introns will create hindrance, especially if you want to express that gene in bacterial cells, because bacterial cells don't have such splicing mechanism. So it's better to isolate your DNA from RNA, or make your DNA from RNA. So you isolate messenger RNA or you isolate total RNA, and out of those total RNA, you use the messenger RNA to make complementary DNA, cDNA. So this is how it takes place. You isolate RNA, and you know in case of eukaryotic RNA, messenger RNA, there is poly-A tail present in the 3' end. So when you isolate this messenger RNA, you do a step which is called as reverse transcription with the help of reverse transcriptase. So reverse transcriptase also cannot initiate a chain. It requires certain primer. So you can make primer by using this poly-A tail, or you can make some internal primer as well. So primer binds to the messenger RNA, and then reverse transcriptase is added, which is basically present in many of the RNA viruses, and that is isolated from source. Or recombinant reverse transcriptases are also available these days, and you need DNTPs because you're going to make DNA. So this reverse transcriptase will make the first strand of cDNA, and then with the partial digestion or with the help of RNase, this messenger RNA can be degraded, and second strand of cDNA can be generated by using DNA polymerase. But nowadays, for PCR, if you want to take this to PCR, you don't have to perform all these steps. None of these steps is required. What you do is you make this cDNA copy, and then even you don't need to degrade this messenger RNA. You take this directly to PCR, and the process is called as reverse transcription PCR or RT-PCR. Because in the first cycle of the PCR here, what happens that these two strands are separated, and from one strand of cDNA, the second strand will be generated by the tag DNA polymerase in PCR. And from then on, the process of PCR will go normally, and from one complete DNA to 2 DNA to 4, 8, and so on. So take-home message is that we isolate messenger RNA, you make first strand of cDNA, and you put it directly to PCR. You omit all these. These would only be needed if you want to make cDNA library. But usually we don't need these days to go up to this level, so you can directly amplify your gene by putting this product in the PCR. As I mentioned, you can make a specific priming, or you can use oligo DT from the poly-A tail, or you can do random priming. It depends nature of your experiment. You can use different primers depending on the experiment you are doing. Only first strand of cDNA is synthesized, followed by PCR in RT-PCR. This is not real-time PCR, this is reverse transcription PCR. Real-time is different, that is quantitative PCR. Now you have isolated DNA or RNA, or you have amplified your gene by PCR. How you are going to check whether your isolation or your PCR product is of correct size? You can do it with the help of agarose gel electrophoresis. Electrophoresis is a technique in which charged species are separated under the influence of electric field. There are different types of electrophoresis such as agarose, polyacrylamide, and there are some other types as well. But here we will discuss agarose gel electrophoresis because it is the most commonly used for nucleic acid separation. So this is a basic equipment of the agarose gel electrophoresis. It has a gel tank, this is a comb, and this is a buffer tank. Here is how you prepare agarose gel. Agarose is a polysaccharide made up of galactose and its derivatives. It is in powder form, and you weigh a certain amount of agarose and add it to a buffer, and you microwave it. And after you microwave it, you pour it inside the gel tank and you place the comb inside it. After a few minutes, you will see this type of gel structure over there, and this comb is there. And the percentage of gel, it depends on the sizes of the DNA you want to separate. If the sizes of the DNA are small, you use more percentage gel. That will create less sized mesh. If you want to isolate larger fragment sizes, then you have to make gel of lower percentage, like 0.8, or even you can go up to 0.5, or sometimes you can go up to 0.3% gel, although it will be very thin gel. But the lower the percentage, bigger will be the mesh size, and larger fragments will be resolved better. So generally you use 1% gel, but if your fragment size is small, like in 100, 200 base and so on, you can use 1.5%. If there are still smaller, you can go up to 2%, 2.5, or even up to 3%. So gels more than 3% are usually not recommended, because the gel becomes so thick that it is difficult to visualize the DNA in the gel of higher percentage. This gel is ready for loading of the samples. Before loading of the samples, you have to add a dye to your samples. This dye will act as loading dye as well as tracking dye. There are two uses of this dye. As a loading dye, it increases viscosity of the sample, and it also gives color to your sample, which is colorless. So it is easier to load this colored sample, and because of increase in the viscosity, the sample remains inside the well. These are the wells which were made due to this comb. So you can easily load your sample in the presence of this loading dye. And second purpose of the dye is the tracking, because when you apply current, the DNA it will run inside the gel, and fragments will be separated. So what this tracking dye does, it shows you how far your DNA of interest is moving inside the gel, because you cannot visualize your DNA. And remember, this loading or tracking dye has no interaction with the DNA. If you see blue color dye running, it does not mean yet that your DNA is running along with the dye. DNA runs independent of the dye. You can track the size of the DNA because you know that, for example, there are two types of tracking dyes. One is bromophenol blue, and other one is xylene cyanol. So this bromophenol blue it runs in the gel along with the size of the DNA of about 0.5 kb or 500 base pair, and xylene cyanol runs along with 5,000 base pairs or 5 kb DNA. So in this way you can track your DNA samples while those are running in the electrophoresis. So you apply a voltage, a current to the to your gel, and this is the gel is dipped in the buffer. You apply your sample after putting the gel in the buffer, and once you turn this on, power supply on, these starts moving, and you can see here the blue color. This is not for the DNA, it is for the dye. So but you if you know the size of your DNA, you can track along with the help of this tracking dye that where your DNA would be, but you cannot be sure until you stain your gel for DNA. So this DNA staining is done by different chemicals, like ethidium bromide is the most common one. This is the general structure of ethidium bromide, and it is a fluorescent dye. It intercalates inside the nucleic acids, and for the fluorescence, you know that there are two wavelengths. One is the excitation, another is the emission wavelength. So excitation wavelength is in this range and under the UV range, ultraviolet radiations. So that's why you need such kind of protection for after this ethidium bromide staining is done. You apply this gel with the UV light. So your eyes should be well protected, your face should be well protected. And the excitation of the ethidium bromide is in the visible range. It is 605 nanometer, and you can see yellow orange colored bands inside the gel. So now these bands are of the DNA. Another important thing is that ethidium bromide is highly carcinogenic. So you should be very careful when using ethidium bromide by using all the protective measures against this chemical and against the ultraviolet radiation. But nowadays there are no more open cameras. You have a certain gel documentation system that is attached to the computer. So usually the bands they won't see in colored form, but they will see this is a computer generated image of the DNA. So you can see these are the DNA fragments, and this is very important to run DNA marker or DNA ladder, or there are different names. This is a standard of the fragments which are of known size. So these sizes are known already. So that's why this is a standard marker. This is called as a marker, or you can also call it as a ladder. Now another important thing is that these nucleic acids always run in the gel in logarithmic form. So here you can see 1857 size band is here, and 1058 is here, and 929 is here, and this is 383. So if you look at the distances and you compare with the size, you would see that this is not linear, rather it is logarithmic. So DNA fragments run logarithmically inside the gel. So if you take a log of these, you will see the difference is quite straight. But here you can see 1857, the difference between these two and these two is not linear, rather it is logarithmic. So you can compare the size of your PCR fragment, like this is here, so you can calculate from here how much size it would be. would have this one, and similarly for the others. Now that you have isolated the gene by using PCR, what to do with that? You have to transfer that gene in some suitable host, such as E. coli, for cloning and expression. But as I mentioned earlier, if you transfer the gene directly to any of the hosts, there are defense systems that will digest that gene into fragments. So you need some vector. You have to transfer your gene in some vector. Vectors have selectable markers, origin of replication, and increased copy numbers. Commonly used vectors are plasmids, cosmids, lambda vector, agrobacterium tumefaciens used for plants, and yeast artificial chromosome. And there are many more vectors that can be used. But here we will discuss plasmids. In one of my advanced lecture, I have discussed other vectors. Plasmids, their nomenclature is written as small p for the plasmid, and then there is a name like UC18 and PBR322 and so on. So some of the vectors are expression vectors, other are just the cloning vectors. And there are some sequencing vectors which have been used by people for sequencing. This is a cloning vector. As you can see, there is an origin of replication and there are selectable markers. These are mainly antibiotic resistance genes like ampicillin resistance or tetracycline resistance. In some of the plasmids, two different antibiotic resistance genes may be present for selection, but in some cases only one antibiotic resistance gene is present. In most of the plasmids, one antibiotic resistance gene is present. Here are the two vectors PBR322, this is its size, and PUC18/19, 2686. Some of these are PUC18, others are PUC19. It depends on the orientation of the multiple cloning site present within the vector. So the three most important features of any plasmid are it must have origin of replication because it has to be replicated independent of the chromosome. So you can see here this is origin of replication, and this is origin of replication. Then it must have at least one selectable marker. Like you can see here, this is a selectable marker for this, which is antibiotic resistance. Here is the ampicillin resistance gene, and here in this case there are two antibiotic resistance genes are there. And then there must be a multiple cloning site, MCS should be present, and that MCS, like this is the MCS, and this is a lacI. This is a part of the beta galactosidase gene that I will discuss later why it is here, and I will also discuss why two antibiotic resistance genes are there in some of the vectors. And this multiple cloning site, basically this is the place where you can cut the plasmid, this circle plasmid, and you can put your gene here. So the property of this multiple cloning site is that it should contain multiple sites, meaning where different restriction enzymes can cut this site. So you have a choice of restriction enzyme that you can use. So there are 8 to 10 or different number of restriction sites present within multiple cloning site. And the most important thing is these sites are unique. These are not present anywhere else in the vector. So here too, multiple cloning site might be present in this or in this. It depends which kind of vector it is. It may be present here or here, but it should be there. And those sites which are present in the multiple cloning site, they are not present anywhere else in the vector, because if, for example, two sites are there, one in the multiple cloning site and other one is somewhere else, what would happen? The plasmid will be divided into two fragments or three fragments or more fragments, which we do not want. We want that this circle should be open from one place. So you can put your gene here. So you can put your gene in the multiple cloning site. And moreover, this multiple cloning site should not be present within the region of replication. There are different enzymes which are required to make recombinant DNA. Here are few of the examples like restriction endonucleases. They cleave double-stranded DNA at specific site. DNA ligase, which joins two DNA molecules or fragments. DNA polymerase 1, it fills gaps in the duplexes. Reverse transcriptase, I already discussed its function. Polynucleotide kinase, it adds phosphate to the 5' end of a polynucleotide. So it is required in some cases. As I mentioned earlier, I'm not going into much detail, but these are the major functions like terminal transferase. It can add homopolymer tails to the 3' end, meaning if you want to add a stretch of any nucleotide, you can use this terminal transferase. For example, if you want to add a stretch of C's at the at 3' end of any DNA, you can use this, and along with this CTP, so these C's will be added over there. Then exonuclease 3 and bacteriophage lambda exonuclease, these help remove nucleotide residues from 3' end and from the 5' end. They have some special function that I will discuss shortly. Alkaline phosphatase, this removes terminal phosphates from 5' or 3' ends or both of these ends, and it also has important function in preparation of the vector. Now starting from the most important enzyme here are the restriction endonucleases. These are mainly present in bacterial cells for their own defense system, and hundreds are purified and now available commercially, and many are in fact recombinant enzymes which are available in the market. And this is the nomenclature that we write first three letters in italic. This first one is the first letter, capital letter, first letter from the genus, and next two are the small from the species, first two letters of the species, and R is the strain and 1 is the number. So this is how this nomenclature goes. Here examples of a few of the restriction enzymes. This is a BAMH1 and CLA1 and so on. And in the restriction site, this arrow shows from where this enzyme cuts. So this is the sequence which is recognized by these restriction enzymes, and this is the place where it will cut. You may notice that in some cases there is a straight cut, like in this EcoR5, it will cut from here. So there will be no overhang in this case, and you will get the ends which are blunt ends. While in these cases you get the overhangs. I will show it more clearly in next slides. But here this asterisk means this place is methylated with CH3 group added at this place in the host bacterium from where these enzymes are isolated, meaning from where these enzymes occur naturally. Why this site is methylated in the bacterial cell? Because this site must be present within the chromosome of the bacterial cell from where these enzymes are isolated. So what may happen that this enzyme will cut its own chromosome. So in order to avoid that, nature has provided methylation function inside the bacterial cells. So there is a modification methylase corresponding to each restriction enzyme present in the bacterial cell that will methylate these of the same restriction sites. So this restriction site which is methylated cannot be cut by that restriction enzyme. Like BMH1 cannot cut if this site is methylated, ACOR5 cannot cut if this site is methylated, this is ACOR1, and so on, different enzymes. Here you can see blunt end restriction site and the sticky end restriction site. So this enzyme PVA2, it cuts straight, and you get these two ends which are called as blunt ends because there is no overhang or hanging DNA. While this ACOR1 cuts here and here and it produces sticky ends, or these are overhangs. Some restriction enzymes produce 5' overhangs, other produce 3' overhangs. Another important thing is that these restriction sites are palindromic. So you can see here like CAG, CTG, CAG, CTG. It is the same sequence from this in this direction and in this direction. So similarly GAA, TTC, GAA, TTC in the bottom strand. So this is palindromic sequence. Moreover, these restriction sites have either 4, 6 or 8 base pairs. These sites are composed of. Now, it is better or it is more convenient if at least one of the end is sticky end while when you are ligating your gene. So this is your gene of interest and this is your vector. When you cut open this vector, from one end is the sticky end, another is the blunt end. So you will easily ligate your gene of interest if it has the same sticky end. Like you can see, same sticky end means it has complementary sequence to this. So it will easily ligate at this place, and on this end it will be ligated with the help of DNA ligase on both sides. Now, another important thing is this vector should not be self-ligated. Especially in case of blunt end ligation, there is a chance that this vector is self-ligated. To avoid that, the phosphates, 5' phosphates from both ends are removed with the help of enzyme alkaline phosphatase, which is mentioned in the last table I show you. And this alkaline phosphatase removes the phosphates from one end. So phosphates are present here, and there are no phosphates are present here and here. So there will be phosphodiester linkage between these on one end here and on other end here, and there will be no phosphodiester linkage. But when you transform this vector inside the bacterial cell, the phosphates which are not present, those will be repaired. Now, there are different methods by which you can screen the recombinant clones. Screening of recombinant clones means how you can find out the bacterial colony which contains your gene of interest. So one of those uses this blue-white screening method, which is the most commonly used and easiest one nowadays. You might remember the lac operon. If not, you can have my lecture on gene expression. Because this lac operon system is used in the blue-white screening. A part of the lacI gene, lacI is the beta galactosidase gene. It is present inside the vector just next to this multiple cloning site. So this is lacI prime, meaning this is part of the lacI gene present here with the multiple cloning site. So you cut your vector open. This is the multiple cloning site from any of the restriction site, and you put your gene at this place. Now when you put your gene at this place, then you transform it into the bacterial cell. So when you transform in the bacterial cell, there is a possibility because this transformation is not very efficient. So a number of bacterial cells won't contain any of the vector. So this is the first situation. So you can see here there is an antibiotic resistance gene is also present there. And second, if the bacterial cell contains your vector, there are again two more possibilities. One is that your vector contains the gene of interest. Another is that your vector does not contain the gene of interest. If your vector contains the gene of interest, that is the recombinant vector, that what you need, what you want. And other one will be the non-recombinant ones. So I will explain it more detail in the next slides, but what you need here is a substrate of this lacI. Natural substrate of lacI is lactose, but here we need a substrate which you can detect by some method. So this X-gal 5-bromo 4-chloro-3-indolyl beta-D galactopyranoside, this is a dye. When lacI acts on this X-gal, this is basically colorless compound, but when this is hydrolyzed by lacI, you get blue color. It gives blue color when this lacI gene breaks it down. Now another important thing, if you remember this lac-operon system, there is a natural inducer of lac-operon is 1,6-allolactose. But here you use IPTG, which is an artificial inducer of the system. And you can see my lecture on lac-operon that it acts with the lacI protein, because lacI is the repressor of the lac-operon system. So isopropyl beta-D 1-thiogalactopyranoside IPTG is used as a artificial inducer of the system. For blue-white screening, this is foreign DNA. It is ligated inside the plasmid vector and then transformed in E. coli cell. There are 4 possibilities here. This is the number 1 possibility, when there is no vector inside the cell. In fact, transformation efficiency is not very high. So there are a number of millions of bacterial cells which remain empty, meaning without the vector. So because we have added an ampicillin to these plates, so all the bacterial cells which do not contain any vector, they are killed, because there is no antibiotic resistance gene here. So if there is no vector inside the cell, it means no antibiotic resistance, because antibiotic resistance gene is there in the vector. Now we are left with 3 possibilities. Now this is another possibility, when this plasmid DNA enters inside the bacterial cell, but there is no insert, meaning no gene present in the multiple cloning site. So in this situation, what happens? We get blue colonies. The second possibility is that insert is present there, but it is not present inside the multiple cloning site, and we get blue colonies once again. And the third possibility is that this is the recombinant one. Our gene of interest is added in the multiple cloning site. In this case, we get white colonies. Now, what is specific reason for getting blue and white colonies? So, in fact, this is the required one. These white colonies are the required ones, because here gene of interest is present and the vector is transformed inside the bacterial cells. There will be many copies of plasmid per cell, and we get this kind of result. So all the blue colonies are without our gene of interest, and all the white colonies, which are actually yellowish type, these are our required recombinant colonies. So we can pick any one of these colony and then use it further to isolate our recombinant protein. Moreover, this one bacterial colony, this is one bacterial colony, this is another one, and this is the blue ones. And one bacterial colony we can see with the naked eye when there are at least 1 million bacterial cells. And the genetic makeup of any of the colony is 100% same. So it means all these colonies have same genetic makeup within the colony, and it may be different from the next colony. This slide is showing mechanism of blue-white screening. This is our vector DNA in which we ligate our gene of interest. So this is the place, multiple cloning site, from where we cut this plasmid circle, and this is lacZ prime. A part of the beta galactosidase gene is here, and this is the antibiotic resistance gene here. So when we do the transformation, two of the options are shown here. The other option where this plasmid does not enter into the cell at all, those bacterial cells cannot grow due to the antibiotic which is added to the bacterial plate. But the bacterial cells containing this plasmid, either it is a recombinant plasmid, this one is the recombinant plasmid, or without our gene of interest, both of these cells can grow because antibiotic resistance gene is present here. So if we are adding ampicillin, this will be ampicillin resistant gene. If we are adding streptomycin, this will be streptomycin resistant gene, and so on. So once this plasmid is inside the bacterial cell, this lacZ component of the beta galactosidase, a part of the beta galactosidase which is called as alpha protein, this is synthesized here, and the other part of the lacZ is present within the chromosome. Due to this reason, we have to select specialized bacterial strain for this experiment, and this lacZ part of it, which is produced from in the host cell, this is called as beta part. So this alpha and beta, when both these combine, it means, and in the presence of IPTG, which is an inducer of the system, as in the operon system, 1,6-allolactose is natural inducer, IPTG is the artificial inducer, and X-gal, which is a dye which is added to this plate. So all the bacterial cells that contain vector but without our gene of interest, those will give blue colonies. Why those will give blue colonies? Because of alpha complementation, meaning this alpha part of the lacZ and the beta part of the lacZ, they will combine together, and they will hydrolyze this X-gal to give blue colonies. So blue colonies are non-recombinant colonies. Now for recombinant colony, this vector will enter into the bacterial cell, and it will produce this beta lacZ protein. But because our gene of interest is present within the lacZ gene, because this is present in the multiple cloning site, so this lacZ prime or alpha part of the lacZ that will not produce alpha protein because of this interruption. So here no alpha complementation take place. What is the result? We get white colonies or yellowish type of colonies, as I mentioned in the previous slide. So this is the basic idea or principle of blue-white screening. This is expression vector. It contains all the signals for transcription and translation. As you can see, there is an operator, promoter sequence here. Then gene encoding repressor, meaning lacI gene, that is also present here. And then this is the multiple cloning site, which actually is present within the lacZ. And this is the ribosome binding site for attachment to the ribosome at correct place so that the first codon of messenger RNA comes in place at the ribosome. So, and this is the selectable marker. At least one antibiotic marker should be there in any of those vectors. And this is the transcription termination sequence, and this is the origin of replication. So when we transform this vector with our gene of interest into the bacterial cells, the bacterial cell will grow and make multiple copies of it, which is cloning, and then it will also make produce recombinant protein, and that recombinant protein may be present inside the cell or it may come out of the cell depending on the nature of the protein and the signal sequence it has. So when we transformation of our vector inside the bacterial cell will provide us with the proteins expressed in the bacterial cell. There are two possibilities, that proteins are expressed inside the cell or proteins are exported out of the cell. In both situations, we have to purify our proteins. If we want to purify from within the cells, of course, competition will be high because more number of proteins are present inside the bacterial cell. But if the protein is exported out, then many of the proteins are eliminated already, and one has to isolate or purify the protein from less number of proteins. So for purification of proteins, because otherwise it is very tedious job like ammonium sulfate precipitation, gel filtration, ion exchange, affinity chromatography, and so on. But if we add some tag to our protein of interest, then we can use this method, which is very easy to isolate and purify the recombinant proteins. Here you need a specific compound, like in this case it is glutathione S transferase is the enzyme which actually act on glutathione. So this is called as GST tag. So this part of the gene glutathione S transferase is present in the vector right next to our gene of interest. For example, this is our gene for target protein, insulin, and this is gene for TST, GST. So what we do is we put these genes as a fusion protein, as you can see here, and when we do the transcription and then translation, those proteins will be synthesized here. So these are recombinant proteins. Now because this GST tag is added here, so what will happen? We have a column chromatography. In this column there is a filling of the glutathione. So this is glutathione is anchored to the medium, and when we add our protein mixture to this column, this protein mixture, remember that this protein mixture contains these fusion proteins with GST tag is attached here. So, and glutathione is present here. So when we add this protein mixture to this column, we start eluting these proteins out of the column. But all the proteins that will contain this GST tag will remain attached to this matrix of the plasmid. So what is the reason for that? Because there is a specific interaction between our the recombinant protein and the glutathione. So when we pass or apply a buffer to this column where these two recombinant proteins are attached somewhere, we change the buffer conditions and start elution. So we have to do it one after the other, and finally our protein of interest, which is attached to this column, is also released from there, and it comes here, comes in the test tube or Eppendorf or microcentrifuge tubes. So we collect different fractions of the proteins here, and in this way we can separate our recombinant protein by using this GST tag. These are some of the commonly used tags for protein purification, and these tags work just like as I have discussed for glutathione S transferase along with glutathione, which is filled in the column. What we have to do is we have to incorporate this tag at the level of DNA. Like we can add 6 histidines. We can add 6 histidines to right next to our gene of interest. And when we want to purify our recombinant proteins, our column will be filled with nickel. So the gene along with 6 histidines will have interaction with the nickel, and all the remaining proteins will be eluted out. And when we change the buffer conditions, our recombinant protein along with the tag will come out of the column. So what to do with this tag which is attached? We just remove the tag with the help of enzyme enterokinase, which cuts the tag from where it is attached to our protein of our interest. There are several techniques which are used. I will just go through very briefly. One is the Southern blotting, although it is not much used these days, but still in some experiments we have to use these. But you will find a lot of research papers using this technique, because before PCR this was the technique which was most commonly used. And this Southern was the name of the scientist. After the name of the scientist, this technique is known as Southern blotting. What we do here is we isolate total DNA or genomic DNA, and then we cut with the restriction enzyme into small fragments. When we run it on agarose gel, because these will be thousands of these fragments will be there, so when we run on the gel we cannot distinguish one from another. So what we do here is we transfer these DNA under denaturing conditions like this alkaline solution. So it means because DNA is double stranded, when we put this gel under alkaline conditions, hydrogen bonding will be disrupted, and single stranded DNA in the form of single stranded, it will be transferred to nitrocellulose paper. So this is our gel, this is the sponge, this is the buffer. So, and these are the paper towels. So with the capillary action, these whole thing will move up, meaning the DNA bands, and because of alkaline solution, they will be single-stranded DNA, no hydrogen bond, so they will be transferred to the nitrocellulose membrane or nitrocellulose paper here. And now we want to check whether our gene of interest is there or not, or if it is there, where it is present. So for that purpose, we use DNA probe in solution form. So this is our nitrocellulose paper, and we add this probe. What is probe? Probe is a sequence of the DNA or RNA which is complementary to the sequence of interest and is labeled with something, maybe with radioactive label or maybe with non-radioactive label, which can be detected. So what happens here is, like, this is the nitrocellulose membrane. We treat with the probe. So because this probe is also single-stranded and the DNA which is transferred is also single-stranded, so this probe will hybridize or will join with the single-stranded DNA, which is complementary to the DNA of the probe. So, and we detect this, we cannot see anything of it on the gel or on the nitrocellulose paper unless we do some autoradiography, meaning we treat it just like X-ray film. So we put it on X-ray film, and after some time we see certain sorts of bands over there. Or if there is sometimes there is a non-radioactive probe, we can use like fluorescence, or we can use chemiluminescence, or different methods of detection. So here I give you a comparison of the southern blotting with similar other techniques like northern blotting. This is for RNA, and northern of course is not the name of the scientist. This northern is compared to the southern is a technique for the RNA. So RNA does not have to be digested with restriction enzymes. We run it on gel, and we do the same procedure as with southern. We transfer to the nitrocellulose membrane and treat it with the probe. And for the western blotting, and this western blotting is for proteins. So here we separate the proteins, and then we treat with antibody. So probe here is antibody. It is not labeled DNA or RNA. It is antibody which is attached with some either radioactive like S35, or it is attached with some non-radioactive material for detection. This is a very good example of the southern blot procedure as applied to one of the techniques of DNA fingerprinting. DNA fingerprinting is a technique which is applied at the crime scenes to identify certain individuals. This is based on polymorphism. Polymorphism is acceptable change or a variation in the DNA sequence within a certain population. Here, this is the earliest techniques used in the DNA fingerprinting is the RFLP, restriction fragment length polymorphism. And we start with the traditional southern blot procedure, isolate the chromosomal DNA, cleave with restriction enzymes, run on the gel, and then transfer to the nitrocellulose membrane and detect the DNA sequence of our interest with the help of a probe. That can be radio-labeled probe, or it can be a non-radioactive probe. So this radio-labeled probe, especially with P32, this has been widely used by many people before this non-radioactive probes, they were emerged. And when this is transferred to the membrane and a probe is attached, it is detected with the help of autoradiography, meaning exposing to the X-ray film, and you can see the DNA fragments here. So in the suspect 1, this is the pattern, and this is the evidence DNA, meaning the DNA which was obtained from the crime scene, and this is the DNA of the victim, and this is the suspect 2. So we wanted to find out which one was the culprit, either suspect 1 or the suspect 2. And you can see very clearly suspect 1 is the culprit because it is the same pattern as the evidence DNA. So basis of this restriction fragment length polymorphism is on the basis of restrictions with the help of restriction enzymes in certain part of the DNA. So all these fingerprinting techniques are developed on the basis of uniqueness of DNA sequence of an individual which is different from the next individual. And this particular technique, when we digest the DNA with the restriction enzyme, that makes a difference, meaning one person may have different number of fragments in a particular region which is identified already by research, and that particular region of the DNA which acts for identification of a certain individual or a certain gene, that is called as DNA marker. So that DNA markers are present. In this case are the sequences where certain restriction enzyme can act differently, making different number of patterns from one individual to the other. And same is seen here, that the restriction pattern of this suspect gives many number of fragments, and here you get fewer number of fragments. No doubt RFLP is the first technique that was used for DNA fingerprinting, but that involves Sutherland blotting, which is quite tedious process. So with the advent of PCR, certain other techniques which are based on PCR analysis have been developed for DNA fingerprinting, including human DNA fingerprints. In human beings, there are short tandem repeats are present in various regions of the DNA. These are identified with the help of research, and these are called as DNA markers. So there are 13 loci which contain these polymorphic short tandem repeats. Tandem means one after the other, and these are short repeats, like maybe 2 nucleotide long. For example, CA is one repeat, so it will be like CA, 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 and so on. There may be like about 100 or so CAs in one case and a different number of repeats in other case. So basically these are the tandem repeats one after the other which are present. And what we do here is, for example, this is the locus where these polymorphic tandem repeats are present. So primers are designed outside this repeat. When we do the PCR analysis, here the example of two different STRs or short tandem repeat markers. So one is longer as compared to the other one. When we do the PCR and you run it on the gel, you find different sized fragments for both of these. So this will give a longer fragment and this will give a shorter fragment. So using this basis, these are the 13 loci at different chromosomes, and these are the repeats. Like in this case, TAGA is the repeat, and this is the repeat length range which is found in different human beings. And total number of alleles which are seen, meaning total number of different variation patterns which are seen in different human beings, is 20 for this repeat, and similarly different like 80 for this repeat. So these are the repeats and these are the repeat lengths. So when we apply, and this last one is to identify whether it is a female or a male. So, and after doing the PCR, you take the DNA sample, you do the PCR using these 13 markers plus the XY marker, and then you run it on a gel. Here you cannot use ordinary agarose gel because sizes of the fragments are very close to each other, so you need high resolution gel, and one of those is the capillary gel. This is the best that can be used. So when we run it on the capillary gel, these PCR fragments, you see this kind of pattern, and this kind of pattern is different in each individual. So none of the two individuals may have same pattern of these PCR fragments out of these 13 loci. Locus means a place on the chromosome. Human Genome Project is one of the biggest projects of human history in life sciences or biotechnology. And this project actually has revolutionized the biological research because it is the sequence of the DNA that is the most important if you want to apply PCR or you want to apply Southern blotting, like you have to make probes in the Southern blotting, you have to design primers for the PCR. So if you have the sequence of whole human genome, and as I mentioned earlier, that the size of the human genome is very, very large. It was very difficult to find out the whole human genome sequence. So in 1990, National Institute of Health of US and the Department of Energy, they jointly started this project, and different labs of the world, they were collaborated. So this is the basic idea, that we digest this human genomic DNA into small fragments, and these smaller fragments, they are ligated into bacterial artificial chromosomes, and it is just like solving the puzzle. And you can imagine the size of the DNA, which is 3 billion base pairs, and which required 80 years to read it once. If you find a job from 9 a.m. to 5 p.m. just to read the human sequence. But the technique we had for the sequencing, that could only sequence a very short part of the DNA, like a few hundred nucleotides could be sequenced at one time, and this is called as dideoxy or Sanger sequencing method. So what they did was, when they cloned this into bacterial artificial chromosome as a vector, and they transformed in bacterial cell, and then they sequenced these back or bacterial artificial chromosomes along with these sequences. They were sequenced, and then when the sequences were obtained, they had to be overlapped just like a puzzle, and finally you get the sequence one after the other. So the project was initiated in 1990, and there was 15 years time with the $3 billion funding. But this project was completed quite earlier, and first draft of the human genome was presented in the year 2000 rather than in the year 2005. So it was 5 years earlier, and later on it was further resolved to a good resolution, and now we have the whole human genome sequence present on the website. So you can see the sequence of any gene using these bioinformatics tools. And bioinformatics science was developed out of it. As biochemistry, it emerged into molecular biology, as molecular biology emerged into bioinformatics. And now, starting from this basically human genome sequence, we have the sequences available for a number of different plants and animals and different species. And having the sequences available, you can easily design any of the project to isolate the particular gene which you are interested in to make more study, or you can isolate the gene to make the recombinant out of that gene, or you can, if you want to get recombinant protein, you can isolate the gene using this sequence data analysis. Transgenic or genetically modified organisms. These are the organisms which have foreign gene introduced into them. So there are examples of animals as well as plants. Here you can see one example of golden rice, where it has a property of beta carotene and extra iron. Similarly, this is an example of Bt corn. Bt is the Bacillus thuringiensis, which expresses a protein which acts against insects. So we don't need pesticide, or we need a little pesticide as compared to the traditional crop in this Bt corn. And similarly, Bt cotton is also one of the examples. And there are certain like soybean resistant to herbicides. Similarly, insect resistant quality traits in soybean, corn, cotton, canola, tomato. So what the idea is, you have a foreign gene introduced into these species, and this is called as genetically modified organism or transgenic. And for the plants, how you make it, we use agrobacterium for this. Agrobacterium has a plasmid which can infect plants naturally. And when it infects plants naturally, it synthesizes certain sort of tumor-like structure, bulging structures are formed. So this information is utilized to make these transgenic plants. Like this is an agrobacterium cell, and this is the tumor-inducing plasmid. You introduce your gene of interest in this tumor-inducing plasmid, and when you transfect this to some plant part like leaf disc, because these plants are totipotent, that leaf when grown in petri plates and you follow this, you finally get full plant, and this is a transgenic plant having a foreign gene. Although it is not as simple as shown here, the actual process is quite complicated, but this is the flow chart how we can make the transgenic plants. Here are very good examples of the transgenic plants. Like this is a tobacco plant having a gene from firefly, luciferase gene. This luciferase is a gene in firefly that helps the firefly to glow at night. So if we transfer this gene in plants, you can see this plant is glowing. Similarly, tomato plants engineered to be resistant against insect larvae, these can be produced by this method. Similarly, glyphosate resistant soybean plants, this is the glyphosate. And the transgenic fish, genes for several variants of green fluorescent protein have been introduced into different strains of zebrafish. So green fluorescent protein is a protein that fluoresces under certain conditions, and then there is a red fluorescent protein, yellow fluorescent proteins. So these fish naturally are not fluorescent, but these are transgenic fish because this GFP or green fluorescent protein and other fluorescent proteins, the genes for this particular protein have been transferred in these fish to make these transgenic fish. Then there is a cloning or cloned animals. This is different as compared to the transgenic. In transgenic or genetically modified organism, only one gene is introduced, or a couple of genes may be introduced into a certain organism. But organism cloning is making a copy of that particular whole animal or whole plant, like or whole human being, maybe his twin sister or twin brother. Like the first one was experiment that was successful was in the Dolly sheep in 1996, and here you isolate the nuclei along with the DNA from the sheep, and you then transfer this into the foster mother. And this whole set of genome is transferred in the foster mother without any fertilization. So the sheep you get here is identical copy of the sheep from where you get this DNA. So if you take DNA out of yourself, the child that will be made, that will be your twin brother or twin sister, because it will be made out of your somatic cells. So there are many different examples of cloned animals, like this success rate has been improved considerably. So these are the first endangered species, and these are the dogs, these are the cats, and you can find many other examples of the cloned animals, which are the animal species which are quite rare or which have a very good demand, that can be cloned to make multiple number of or copies of that particular animal. And finally, if we look at the list of recombinant proteins, you will find a huge number of proteins. These are just a few examples. As you can see, human insulin for treatment of diabetes, it is now in recombinant form. So there is a long list of hundreds of proteins which are coming out as recombinant proteins. So with this, I end my lecture. So this was an overview, just a brief overview of the recombinant technology. So for details of each of these techniques, I will have separate lectures on different techniques. Thank you very much.